Our next speaker is Dr. David Furman. He directs the AI and bioinformatics platform here at the Buck Institute, is head of the Stanford 1000 Immunomes project, and is the founder and CSO of Cosmica Bio, a company that is enhancing human resilience for longevity and deep space exploration. And he's going to tell us about useful biomarkers and aging clocks.
All right, thank you for that intro. I don't have very good news. I'm going to talk about things that were discussed previously, but I don't have that beautiful British accent that Madeleine and Ranjan have. So thank you, Christine, for inviting me. It's really good to present here at home. And I'm going to go quickly through the first set of slides, because most of you should know this background. And then I'm going to focus mostly on the last two bits here. But I'm going to explain a few things that may be important in the context of health span, which is what we're trying to expand here at the Buck Institute with the research we're doing. So you probably all know that there's a difference between lifespan and health span. We're trying to expand and extend health span as much as possible to achieve also healthy lifespan and increase that by default. And how do we do that? By using wellness and prevention as well as diagnostics and therapeutics. And the ultimate goal is really to extend the quality of life, the wellness-adjusted life years, and compress morbidity. That idea Freisa put out in 2008. And not only because we want people to live better, but also to reduce the burden in the economy. 90% of the annual budget in healthcare goes for chronic diseases associated with older age. So if we can tackle these, that's $4.1 trillion in a year only in the US. So to that extent, I'm really, really delighted that Nathan Price and Lee Hood join us in this effort to create the Center for Human Health Span here at the Buck Institute. And we're now, with John Newman and Brianna Stubbs, running actual clinical trials to improve a health span and extend lifespan as well. So this is just a kind of a big picture of things that change with age. Many, many, many things change with age. And I'm not going to go into the detail, but things can change linearly as well as non-linearly, as Ron alluded to, John, earlier today. And the claim that aging is complex, I think, is terrible. It's a bad claim because it implies that we're not understanding it well. And I think we have made terrific progress over the past 10 years to understand aging a little bit better. And honestly, it's no more complex than the function of a cell, right? There are different layers, and I think Luigi Ferrucci published this paper in 2018 to really illustrate that there are different layers, this hierarchical process where molecular and cellular changes happen earlier in time, and then tissue aging and systemic aging happen later in time. With things like disability, intrinsic capacity, loss of function, and frailty. So biological hallmarks of aging, we all know this is the very early things that change with age at the molecular and cellular level. And this is phenotypic and functional aging. This is a collaboration we're doing with Joel Kramer at UCSF, looking at gray matter volume that changes with age, goes down with age. This work that Nico Markov has done in my lab. And you can see that there's a pre-mild cognitive impairment in yellow. That's already functional decline, right? So here we're combining phenotypic aging with functional aging in one plot. So the progression of aging goes biological, phenotypic, functional, even before a disease kicks off. How do we measure that first layer of biological aging? All of you know the epigenetic clocks. There's more and more development in the biomarkers of aging as well as clocks. And I wanted to summarize this in this table and tell you what we think is, or I think rather, is my favorite one, is really inflammatory clock, because it can give you a really good intervention guidance, as opposed to more standard first generation clocks. So here we see the accuracy in predicting chronological age. It differs in different types of data sets. And then the known biology, we don't know really how to action it very well with epigenetic clocks. That's different from cellular clocks and proteomics clocks. And the interventions, if we really understand the proteins that are circulating and the organs that we want to target, that gives clinicians and consumers a much more focused way of intervening. Is actionable. So on that front, we started the 1,000 Immunomes project at the Stanford School of Medicine to identify chronic inflammation biomarkers, and we built a clock for inflammation. And that tracks with many different clinical endpoints like multimorbidity, frailty, 7 years before it happens, with immune function is a surrogate of immune aging, and heart health, so this is ventricular remodeling. So the higher the inflammatory age, the worse the phenotypes overall in this 1,000 Immunomes cohort, as well as an independent cohort of exceptional longevity individuals that we gather from a collaborator in Italy. We can also predict mortality. And we brought that to the marketplace. We're now commercializing this through the companies called Edifice Health, mostly through longevity clinics, wellness centers, and functional medicine physicians. So I think there's good news for everybody who is looking to focus on the biology of aging. We now know that the WHO has recognized age-related decline in intrinsic capacity as a disease, as part of the International Classification of Diseases, ICD-11. And we're now measuring functional aging at large in collaboration with Dr. Bruno Vella in Toulouse, where we have 1,000 people that have been followed for about 4 years now, and the project will last for 10 years. We secured a large amount of money from the European governments, and to measure cognitive status and a number of different aging phenotypes, as well as multi-omics, right? So we are now using baseline multi-omics measurements to predict different functional outcomes with aging. So this work was done mostly by Matias Fuentealba in my lab, who's here in the room, where he built a proxy, methylation-based clock for functional aging. This is not trained on chronological age, and then as first-generation clocks do, but this is trained on function, like second-generation clocks. And so you can see that there is sexual dimorphism in the rates of change in these different domains of intrinsic capacity, and we built that predictor, which is noisy, and I love noisy clocks, because that gives you the opportunity to look at the biology in this axis, right? You have the predicted intrinsic capacity, and you have the real intrinsic capacity, and almost no CPG that were selected by this new clock overlaps with first and second generation clocks. The intrinsic capacity clock is able to predict immune health and mortality. We did a number of different association testing here with the Framingham Heart Study, which was not used to train the algorithm. Remember, it was trained in the Inspire cohort in Toulouse, and then we're validating in the Framingham Heart Study, and we can see that those intrinsic capacity clock metrics are associated with lower naive CD8 T cells and exhausted CD8, CD28 expression, in looking at the RNA sequencing. That seems to be very important for T-cell activation and so on and so forth. And then we're also predicting all-cause mortality as well as cardiovascular disease-related mortality, much better than any other first and second generation clock. We can also use gene sets based on LLMs that are associated with different hallmarks of aging and ask the question, is my intrinsic capacity clock related to hallmarks of aging? And the answer is mostly for chronic inflammation, cellular senescence, and to a less extent, to altered intracellular communication stem cell exhaustion. We can also associate this high intrinsic capacity with health overall, with exposome, with the idea of lifestyle choices as well as inflammation. You can see that the inflammatory age clock that I alluded before is the number one top negative association with our intrinsic capacity clock, and things like functional measurements like hand grip and lung capacity are positively associated with higher intrinsic capacity overall. And then you can see here that there are certain things in your foods that drive higher or lower intrinsic capacity, like flavonoids are all positively correlated. And then fatty acids coming from animal sources, like omega-3 fatty acids, are driving positive intrinsic capacity, whereas the other fatty acids, like omega-6, are negative. So I'm going to jump into a really important breakthrough study that also Matthias Fuentealba has spearheaded in collaboration with Eileen Kremins at USC. And that is to use the UK Biobank as well as the health and retirement cohort to build what I call the third generation clock, which is we're using trying to predict ICD-10 chapters, and these are different diseases that people died from, using clinical lab measurements first, and then create a metric that we can predict using multi-omics, proteomics, metabolomics, DNA methylation patterns, and transcriptomics. And so how do we do that? I'm just going to jump into the important part right away. This is just to show you a few differences between first and second generation clocks. For the most part, I would say the important piece here is first generation clocks are focused to train on chronological age, whereas second generation clocks are mostly focused on health outcomes, predicting mortality, predicting different diseases. And really the usefulness of first generation clocks is questioned, largely questioned, because we don't know really, these are high level acceleration of your epigenetic clocks doesn't mean much as a simple overall view of your aging. But if you have now a clock that will tell you, well, you have the brain acceleration or your heart acceleration, clinicians and consumers can guide the interventions that are out there for the heart or lung or brain. And so Ron John alluded to this work from Mike Schneider as well as Tony Wiscorey at Stanford. There's a new study that was published only a month ago or a couple of months ago by Aaron Segal in Israel at the Weizmann Institute. All this is second generation clocks. Train on diseases, train on mortality. How can we do better than these people? Very difficult, very different. And so what are we trying to do here is to use a system-specific focus, and for that we use ICD-11 chapters, ICD-10 chapters like infectious disease, endocrine, mental, nervous system, circulatory, respiratory, etc. Then we also are using Cox penalized regression. This is a method that was developed early on in the early 2000s to analyze gene expression data in an unbiased fashion that can select features in a multi-dimensional space that are associated with, let's say, a high accuracy of your Cox proportional model in predicting mortality, for example. And then we use the UK Biobank, which is an ongoing study. We're going to have more data in the future, as well as the health and retirement cohort that has the multi-omics. So again, I mentioned this is work done in collaboration with Eileen and Arpa Wong at USC. And what I'm showing you at the top of this slide is how the PhenoH clock was conceived in the first place. What's the method to derive the PhenoH clock? So they use also blood chemistry, cell count, and age to predict mortality, all-cause mortality. And then they created PhenoH, right? That's your predictor. And with that, you have epigenetic data that you can use to predict, based on ElasticNet, the PhenoH. And that's the DNA methylation PhenoH that True Diagnostics is commercializing. So what did we do here? We are using similar features. We added gender or sex to predict, instead of all-cause mortality here, we're predicting ICD-10 chapter specific mortality. Then we're transforming the data to represent that in an age component, in a kind of a disease age, we call it. And then we use metabolomics, proteomics, as well as epigenetics and transcriptomics to predict the omics disease age that this didn't come very clear. But what I'm showing you at the top of this slide is the 500,000 people and 10,000 people from the Biobank and Health and Retirement cohort respectively. And the different disease chapters that you see in the ICD-10, circulatory, digestive, urinary, infectious, mental, metabolic, and so on. And what you can see is that the correlation with age, when you build the disease age metric, is generally pretty good, right? And for two studies are very, very similar. The correlation with age is so similar. You can see here, it's almost one. It's very, very good in terms of the validation that we have in a study over the other. And then this is the C index, which is the prediction of the accuracy of the model at predicting mortality due to those causes. And you don't see here, but it's all above 0.75, which is really strong predictor of mortality in those cases. When you look at specific diseases, they're all in the system that we're predicting disease age acceleration. Okay, so that's shown here. The disease age acceleration, for example, for angina, congestive heart failure, and heart attack is always circulatory. That's the largest age acceleration that you see, and that's also true for other conditions and diseases. When you look at people that are still alive versus those that died over the course of the UK Biobank study, there are major differences in this. And people with circulatory issues will also have the tendency to have respiratory issues, metabolic, musculoskeletal. And for musculoskeletal, there are such high chance of people developing nervous system and digestive issues and so on. This is a hazard ratio for the cause-specific mortality associated with other systems. And then last, we looked at extreme phenotypes. So we used 2 standard deviations from the mean, and then we're asking how many people, or what's the frequency of individuals that are suffering from disease age in one system versus 2 versus 3 versus 4. That's listed here. And then the association between extreme phenotypes and age-related risk. And with this, I'm going to try to wrap up because I'm in a yellow here, light, and that means I need to wrap up. So anyway, so when we take the massive amount of data that was collected in the UK Biobank and in the Health and Retirement cohort with 10,000 people, 500,000 people, and 1,500 features for proteomics, metabolomics, and then 16,000 genes and so on, we're now connecting what I would like to say the largest disease omics mapping that exists to date. And what I'm showing you here is the correlation with age. So proteomics does better at predicting disease age as it relates to age. And we're looking at the C index, which is the concordance index to predict mortality, with an average of, let's say, 0.75, which is actually pretty good. So I'd like to just summarize. I touched upon inflammatory age as a novel marker for inflammaging. I didn't show you that, but it is sensitive to an intervention. Intrinsic capacity clock, this is clinically relevant, and disease age as the largest disease omics mapping resource, and this is being built in this website that you can see at the bottom of the slide. And with that, I would like to thank my team and thank you for your attention. Questions?
When it comes to interventions, you said it's sensitive to interventions. I know you didn't put any data up there, but that's really what we're looking for as clinicians. What kinds of interventions did you look at, if you have any information to share with us?
Yeah, for the inflammatory age clock, we worked with a corporate partner to test whether the inclusion of a high avenanthamide meal changes inflammatory age. And that is taken 3 times a day for 2 months. And as early as 2 weeks, we saw a reduction in protein called eotaxin and inflammatory age overall in people with high inflammatory age. It's really difficult to improve the biological aging in individuals that have good figures to start with. So if you are in the kind of an high inflammatory age or high biological age by other means, there's a larger delta that you can achieve. In our case, the significant changes we observed in people with high inflammatory age at baseline.
Thank you.
It's a paper that has been published, so you can. Yeah. No, that's a great question, and the answer is yes. I think 3rd and 4th generation clocks will include not just multi-omics as a concept, but also incorporation of longitudinal data, which is what we are lacking here. And so the INSPIRE is in year 4th. We published a paper only on baseline, and we're now incorporating the longitudinal data to model a better and more accurate clock.
Wonderful presentation. You're talking about inflammation a lot. What is your forecast for GLP-1 medications regarding inflammation and perhaps morbidity and mortality rates in the future?
I have no clue. I do not know of any research showing that GLP-1 drugs will lower inflammation as far as I understand from a aging perspective. It may improve acute markers of inflammation like IL-6 or TNF-alpha or CRP, but I haven't seen data showing that it may improve age-related inflammation. No, we haven't had the opportunity to look at data on our own cohorts. Yeah, we don't have the data.
Thank you for your talk. Right here.
Yeah, hi.
Hey. I was wondering, as you're looking for different systems and their correlation with dysfunction in one system correlating with dysfunction in others, did you find a single system that was like a canary in the coal mine where early dysfunction in this system predicts that subsequently you will be one system affecting several others?
Not that I recall. Matthias is right here. But what I do remember, one of the analyses that he has done is the aging of the gut seems to precede the aging of the brain by about 10 years. And whether or not that is causal, we don't know, because there's no longitudinal follow-up to make sure. But there's a connection there. Yeah. Yeah.
I'm interested in looking at possibility, if there anybody, you or anybody else has looked into gut microbiome diversity and how that affects in immune age.
Yeah, of course. At the Center for Healthspan here at the back, we're looking at microbiome at large in multiple studies. There was a study published not long ago by Nathan Price looking at microbiome and metabolites and how that accelerates aging and the relationship between different genomes and families of bacteria there. In my own hands, larger diversity corresponds to lower inflammatory age. That's in the 1,000 immunomes cohort. So a breadth of different bacteria there will give you protection. And that's kind of the general understanding by looking at Shannon diversity. But that's a very rough approximation. Yeah. Only in older adults, not in young people. Yeah. Yeah, there's one question over there.
There is a question here first. Sorry, on the same vein as GLP-1s, I'm just interested about rapamycin and whether you have seen that impact. And there's been some recent studies looking at epigenetic markers with it. But yeah, I'd love to hear you speak to that.
Yeah, again, we don't have the analysis done yet by medication, but I think that's a really good idea that my team is picking now to include in the manuscript. I think the understanding how our disease age and our intrinsic capacity age are changing with different medications is absolutely key, but we don't have the data yet.
No more clocks.
Performance in predicting disease or predicting chronological age or what exactly?
I assume it's predicting lifespan and health span in a way that's performant enough.
Yeah, it's an interesting question. We're not particularly focused on one number, but what I can say is that the accuracy of the clock, when you correlate that metric with age, has zero relationship with how that clock is able to predict health outcomes. So let me just show you real quick. That was right here. One sec. So here you can see that the noisiest clock, which is digestive and metabolic, are not necessarily giving us bad predictions for diseases. So metabolic is in the top 3, and you have a correlation with age of 0.54. Whereas the most accurate clock, which is mental here for both the health and retirement cohort and the biobank, it gives you a prediction of the disease chapter only in the 4th level. So there's no correlation between the accuracy of the clock and the ability to predict health outcomes, which was, to me, very surprising and really very pleasant to see that, because most of the clocks we use are pretty noisy, and I really like that, as I said before. Yeah, thank you.